This video demonstrates a protocol for the isolation of Hoechstlow CD45-negative lung mesenchymal stem cells. Tissue-resident mesenchymal stem cells, or MSCs, are important regulators of tissue repair or regeneration, fibrosis, inflammation, and angiogenesis. First, the lungs are dissected from a sacrificed mouse. The lung tissue is digested to obtain a single-cell suspension. The cells are stained with Hoechst dye and CD45 and sorted by flow cytometry to obtain Hoechstlow CD45-negative lung MSCs. The lung MSCs are then expanded in culture, and a colony-forming assay is performed to assess the MSC differentiation capabilities. Further studies can then be performed to examine the role of MSCs during tissue homeostasis and disease.
The method we show here can be used to isolate either mouse or human lung mesenchymal stem cells and can be applied to the study of lung diseases such as pulmonary fibrosis, pulmonary hypertension, and COPD.
Generally, individuals new to this method will struggle because of the variety of methods available for tissue preparation, as well as flow cytometer setup, appropriate controls, and analysis.
Visual demonstration of this method is critical since it is difficult to describe the proper technique for mincing and digesting the lung tissue for optimal viability of cells with words alone. Also, the flow cytometry instrument setup and analyses are best communicated through demonstration.
Begin this protocol by removing the lungs that will be used to obtain mesenchymal stem cells from a sacrificed adult mouse. Use a small pair of sharp scissors to cut the ribcage laterally on each side of the mouse. Open the chest cavity, then remove the diaphragm. Insert a 10 milliliter syringe with a 20 1/2 gauge needle into the right heart ventricle and gently depress the plunger to perfuse it with 3 to 5 milliliters of PBS, flushing the blood from the lungs. Next, using scissors, snip off the trachea and large bronchi and discard them. Then using forceps, pick up the small white lung lobes and place them in a petri dish filled with Hoechst buffered saline, or HBSS. Then remove the heart and separate the lung lobes. Repeat this process for all of the mice in the study. Transfer the lung lobes to the lid of the dish. Moisten the tissue with just enough HBSS to keep them from drying out. Then, using a disposable scalpel, mince the lung lobes. To obtain bone marrow to use as a staining control for flow cytometry, use a pair of sharp scissors to cut at the ankle and top of the femur. Place the limb into a new petri dish. Then cut off the knee, leaving a linear piece of femur and a second piece containing the tibia and fibula. Using forceps, pull off the muscle to reveal the tibia, fibula, and femur. Now take a 5 milliliter syringe with a 26 1/2 gauge needle and insert the needle into the bone marrow opening at one end of the tibia. Hold the bone with the other end directed into a 50 milliliter conical tube filled with 15 milliliters HBSS and depress the plunger to dispense HBSS and flush bone marrow into the tube. Repeat this process with the fibula and femur. Stain the bone marrow with Hoechst 33342 dye at a final concentration of 5 micrograms per milliliter in supplemented high glucose DMEM. Next, a single cell suspension of lung tissue will be generated from the isolated tissue. Place each lung in a tube containing pre-warmed 0.2% Worthington type 2 collagenase prepared in sterile HBSS. Then immerse tube in a 37 degrees Celsius water bath and incubate for 30 minutes. Following the incubation, use a 10 milliliter pipette to triturate the sample until the digest flows easily through the pipette, approximately 10 repetitions. Incubate for an additional 15 minutes at 37 degrees Celsius to complete the tissue digest. Once the digest is complete, dilute the cell suspension with HBSS and use a 5 milliliter pipette to triturate again to disperse any residual tissue fragments. Next, to remove undigested tissue fragments, pour the suspension through a 70 micromolar cell strainer into a 50 milliliter conical tube. Add suspension a little at a time to allow sample to flow through the cell strainer. If undigested tissue fragments completely block the flow of the cell suspension, pour through a new cell strainer into a new tube. Pellet the cell suspension for 10 minutes at 450 RCF. Following the spin, decant the supernatant and gently resuspend the cell pellet in room temperature red blood cell lysis buffer. Incubate at room temperature for 5 minutes. Then add an equal volume of HBSS to inactivate the lysis buffer. Pour the cell suspension into a 40 micromolar cell strainer to remove debris and cell aggregates, collecting the flow-through in a new 50 milliliter conical tube. Next, using a hemocytometer, determine the cell numbers for both the lung and bone marrow samples. Record the concentration and total volume of the cell suspension. Then, pellet the single lung cell suspension by centrifugation at 450 RCF for 10 minutes. To stain the cells, resuspend both the lung and bone marrow cells at 1 × 10 to the 6th cells per milliliter in pre-warmed supplemented high glucose DMEM. To stain the DNA, add Hoechst 33342 dye to a final concentration of 5 micrograms per milliliter. Next, mix the cells by gentle inversion and incubate in a 37 degrees Celsius water bath for exactly 90 minutes. Then centrifuge for 10 minutes at 450 Gs at 4 degrees Celsius. After the spin, proceed with staining of the lung and bone marrow tissue with anti-CD45 and propidium iodide in preparation for analysis by flow cytometry. Once the cells have been stained, store the tubes on ice, protected from light until flow cytometry analysis is performed. Prepare for flow cytometry by ensuring that the instrument's lasers are aligned and that it is set up for cell sorting. The instrument used must be equipped with blue 488 nanometer, red 635 nanometer, and UV 350 to 355 nanometer lasers, with 2 detectors on the UV laser path with blue 45/65 and red 675/50 filters. In addition, the UV red detector must have high sensitivity for the red signal, and the instrument must be equipped with a chiller unit to maintain the sample at 10 degrees Celsius. In the instrument software, set up histogram plots to display linear forward versus side scatter, a doublet discrimination plot appropriate for the instrument, a histogram plot of PI using a log signal, a red versus blue Hoechst using linear signals, and a histogram plot of CD45 APC using a log signal. Once the instrument is ready, place the stained bone marrow sample into the loading port of the instrument. This sample is used as a staining and instrument setup control. Begin collecting events and adjust the forward and side scatter linear signals to clearly visualize the cell population. The cells should reside approximately in the lower center of the plot with forward scatter as the x-axis. Since the nuclear stain PI is membrane impermeant, it will be excluded from living cells. Draw a gate region on the PI-positive dead cells in the PI histogram. Instruct the instrument software to color the dead cells in this gate red. It is helpful to use the color-evented dead cells as a navigator to identify the host G0, G1 population. The PI stain is also excited by the UV laser, and the majority of the dead cells should be off-scale on the right side of the red versus blue Hoechst fluorescence dot plot. The G0, G1 population of the bone marrow should be seen as a tight cluster of events in the red versus blue Hoechst plot. Adjust the detector voltages to place the G0, G1 cluster in the upper right of center on the plot. A portion of the S phase and the entire G2 of the cell cycle may be off scale on the upper right of the red versus blue Hoechst plot. The side population, Hoechstlow, will be seen trailing from the left side of the G0, G1 cluster down to the lower left corner of the plot. Now draw a gate around the cell cluster in the FSC versus SSC plot. The singlet population identified in the doublet plot, and the live cells in the PI histogram. Then assign the Hoechst dot plot to display only these gated populations. After gating the Hoechst plot, draw a region around the side population. Assign this region as a gate to the CD45 APC histogram along with the light scatter, singlet, and live cell gates. Now that the system is set up, remove the sample from the loading port and place the lung sample in the loading port. The instrument settings should not need to be adjusted again for this sample. Begin collecting events. On the red versus blue Hoechst plot, the G0, G1 cluster for the lung sample usually appears more elongated in the x-axis direction and resembles the tilde symbol on a keyboard. The side population gate Hoechstlow should be in a similar position to that set for the bone marrow sample, although slight adjustments up or down may be necessary. To ensure tight resolution of the side population, maintain a low pressure differential during sorting by ensuring that the sample flow valve isn't opened too far. Next, to isolate MSC, place a 96-well collection plate in the sort chamber and set up the sort gates on the CD45 APC histogram to separate positive and negative populations. Finally, collect the cells as either a mixed population into a tube or single cells to isolate clones into a 96-well plate. Following collection of the lung MSC by cell sorting, the cells are plated in 30mm dishes using AMEM supplemented with 20% FBS. Freshly sorted cells appear small, round, and bright. After approximately 2 to 3 weeks, colonies with a mesenchymal phenotype become evident and proliferation is more obvious. For each cell preparation, evaluate the ability of lung MSC to differentiate into traditional mesenchymal lineages of osteoblast, adipocyte, and chondrocyte, and their cell surface expression of accepted mesenchymal markers. Perform cytogenetic banding analysis to confirm the absence of gross chromosomal abnormalities. Following isolation by flow cytometry and generation of single-cell clones, a colony-forming assay is performed to characterize the MSC cells and the differentiation capabilities. Begin with cells diluted to 3 × 10⁴ cells per milliliter in complete mesenchymal medium. In 100mm dishes, perform serial dilutions to 6 × 10⁴, 3 × 10⁴, 1.5 × 10⁴, and 0.75 × 10⁴ cells per dish in 10 milliliters of media in triplicate. Then place the plates in the incubator. After 10 days of culture, pour off the medium and rinse the cells with PBS. Then fix them by adding 100% methanol for 5 minutes at room temperature. After 5 minutes, pour off the methanol, then to detect colony formation, add 0.4% weight by volume GMZA, diluted 1:20 with deionized water. Incubate for 10 to 15 minutes. Then pour off the stain and rinse with deionized water. Allow samples to air dry and quantify the number of colonies containing greater than 25 cells per colony using an inverted microscope or visualization. Colonies are large and comprised of a few hundred cells as shown here and display a mesenchymal phenotype. MSC were isolated as shown in this video and plated in 96-well plates. Simulated Growth-arrested, CFSE-labeled, antigen-presenting cells were then added to the lung MSCs. The proliferation of isolated MSCs was then measured as the decrease in mean fluorescence intensity of CFSE compared to the background, which was CFSE-labeled T cells alone. As shown in this figure, in the absence of lung MSC and presence of antigen-presenting cells, APC plus/minus ovalbumin CD40-positive effector T cell demonstrate a decrease in CFSE intensity, which indicates proliferation, circled in red. CFSE fluorescence intensity of the T cell membrane decreases stoichiometrically with every cell division, i.e. 1/2 with every division. In the presence of lung MSC, APC plus/minus OVA CD40-positive effector T cells demonstrate no significant change in CFSE intensity, which indicates a lack of proliferation.
After its development, this technique paved the way for researchers in the field of lung stem cell biology to explore the role of mesenchymal stem cells in lung diseases such as pulmonary arterial hypertension and pulmonary fibrosis.
Once mastered, this technique can be done in approximately 5 hours.
Following this procedure, other methods like differentiation assays can be performed in order to answer additional questions, including whether or not the putative lung mesenchymal stem cells exhibit appropriate multilineage differentiation potential to bone, fat, and cartilage.
About the method. This method initially used to identify BM hematopoietic cells to isolate a specific population of resident lung MSC. The recent definition of this population in vivo in both murine and human pulmonary tissue facilitates the development of a therapeutic strategy directed at the rescue of endogenous cells to facilitate lung repair during injury and disease. The ability to repeatedly isolate this characterized population allows the further study of the biological importance and role of the lung MSC during tissue homeostasis and disease. Thank you for watching. Subscribe if it is useful.